Bismillahirrahmanirrahim. In this lecture, we are going to talk about pathogenic Clostridia. Objectives: the general characters of genus Clostridia, the most pathogenic member of genus Clostridia, morphology, various factors, transmission, and pathogenesis of its diseases, and finally should know the outlines of diagnosis, prevention, and treatment. Genus Clostridia are large gram-positive bacilli. They are obligatory anaerobes. That means oxygen is toxic to them. They found in the soil, and they are normal inhabitants in the GIT of human and animals. They are spore-forming bacilli. Most Clostridia are motile by means of pretreacherous flagella. Many members are saprophytes, and the other are pathogenic. The spores of these bacilli are wider than the bacillary body, giving a swollen appearance. This swollen appearance resembles a spindle, and hence the name cluster, that means spindle in shape. The shape and position of spores are varies in different species and can be used as a useful method for identification. These spores may be central, terminal or subterminal, may be spherical or oval in shape. The most pathogenic Clostridia we will study are, first, Clostridium tetani, causing tetanus, Clostridium welchii, or Clostridium perfringens, causing gas gangrene and food poisoning, Clostridium botulinum, causing botulism, Clostridium tetani, causing tetanus. Tetanus means being taut, extreme spasm, which is a good expression for the disease. The bacilli here is gram-positive, motile bacilli with terminal bulging spores, giving the drumstick appearance. This bacilli is highly resistant to heat and other chemical disinfectants. The bacilli is a normal inhabitant of the gastrointestinal tract of human and animals, and in the soil, due to the presence of oxygen, which is unfavorable condition, the spores is formed, and these spores remain viable for many years. When these spores enter inside the body through a trauma or even dirty or clean wounds, they germinate again to the bacillus form and cause tetanus, which is characterized by spasm. The spasm begins in the facial muscles and then spread throughout the body, causing severe muscular spasm and contractions. These spasms are characterized by sudden, powerful, long-lasting, and painful spasm. It may result in muscle tear or bone fractures. Sometimes the spasm is localized around the area of injury, and this is called local tetanus. Other condition called neonatal tetanus, where the newborn acquires the tetanus from the mothers through the cutting of umbilical cord by non-sterile instruments, while newborns from immunized mothers are protected. Pathogenesis: when the spores enter the body via the wound or trauma, as we said before, It germinates to give the bacillus form. The Clostridium tetani bacilli produce a potent neurotoxin. This toxin called tetanospasmin exotoxin. Shortly, this toxin is carried intra-axonally to the CNS, where it binds to the ganglioside receptors and make blocking to very important inhibitory mediators, glycine and GABA. In normal condition, these inhibitory mediators are responsible for the relaxation and elongation of muscle fibers. But when the tetanospasmin blocks the release of such mediators, it results in excitatory neurons and extreme muscle spasm. The clinical signs of tetanus are triad. First, teresmus or lockjaw, mild to severe spasm of lower jaw master muscles. Then, rhizus sardonicus, or this characteristic facial expression due to facial muscle spasm. When the spasm is generalized all over the body and all the muscles of the body are spasmed simultaneously, this is called obstotenus. This clinical photo shows an infant with generalized tetanus, obstotenus, and rhizus sardonicus. Laboratory diagnosis depends primarily upon the clinical signs, severe muscle spasm and rigidity. Clostridium tetani bacilli will not be isolated from the wound. It's very difficult to be isolated. However, in case the sample is wound exudate, the stent film reveals gram-positive bacilli swollen at one end due to the terminal spherical projecting spores that may appear in the tissue. Culture and blood agar anaerobically. Treatment of tetanus requires attention to a number of important factors. The stimulation of the patient should be kept to a minimum. Even the light of the room should be kept minimally. The tetanus immunoglobulin, which is the anti-tetanus antitoxin, should be administered intramuscularly immediately at a dose of 500 units to neutralize any toxin that has not already entered to the CNS. It neutralizes the toxins that not already entered to the CNS. Diazepam is extremely effective to sedate the patient and to induce muscle relaxation, cleaning of the wound, and treatment with antibiotic, penicillin, or metronidazole is effective. Prophylaxis. You should know that the tetanus as a disease is entirely preventable disease. Entirely preventable. That means adequate immunization will prevent the occurrence of the disease. And this immunization is present in the immunization schedule here in Egypt, where the children take the tetanus toxoid in combination with diphtheria toxoid and pertussis in what's called DPT vaccine. The children take it 3 doses intramuscularly at 2, 4, and 6 months in Egypt, in the immunization schedule in Egypt. A booster dose is given at school entry. Also, pregnant women can take a booster dose to guard against transfer of infection during labor. Passive immunization by giving the intramuscular injection of tetanus antitoxin to the wounded persons without history of vaccination. Now we are going to talk about Clostridium welchii or Clostridium perfringens. It is a large gram-positive bacillus, spore-forming. The spores are oval, subterminal, and non-projecting. It is capsulated and non-motile bacilli. It is one of the most growing bacteria known. Its replication time is as short as 8 minutes. The dose of Clostridium perfringens, including a number of powerful toxins. The most important of these is alpha-toxin or lysosinase or phospholipase that is associated with severe toxicity, hemolysis, and myocytes in a condition called gas gangrene. Also, spreading enzyme as collagenase and hyaluronidase worsens the case. Another virulence factor responsible for other condition is called neurotoxin, which is responsible for the food poisoning by disruption of ion transport and resulting in watery diarrhea. Clostridia causing gas gangrene. Shortly, two types of Clostridia causing gas gangrene. Saccharolytic type, which ferment carbohydrates producing acid and gas, and proteolytic type, which digests the proteins with a blackening and bad smell. The saccharolytic type, example for saccharolytic type is Clostridium welchii or Clostridium perfringens. Example for proteolytic type is Clostridium histolyticum, and mixed type called Clostridium bifragmentans. This gangrene is a very bad condition. It begins with the deep lacerated wounds, car accident or war wounds, or sometimes in operation in the abdomen and lower extremities that is contaminated with the spores. When the spores enter inside the body, it germinates to produce the powerful alpha-toxin. As we said before, this alpha-toxin causes direct damage of the cells and swelling up of the cells, and rapid, massive destruction of muscular and soft tissues, resulting in tissue death or gas gangrene. And this is a clinical photo showing the condition. This condition is accomplished by generalized toxemia, shock, and death. The second condition caused by the Clostridium perfringens is the food poisoning. It is due to the ingestion of contaminated warmed meat dishes containing the spores. The incubation period is up to 18 hours. The spores are germinated inside the gut and release the enterotoxin. The symptoms of the condition is abdominal cramps, water diarrhea, and vomiting. But it is a self-limiting condition. Through 1 to 2 days, it is self-limiting and we are not in need to take antibiotics. But keep the individual rehydrated. Laboratory diagnosis made on the basis of clinical presentation also. The sample in case of gas gangrene is a wound exudate. The standard film reveals gram-positive capsulated rods, non-motile. However, the spores here are rarely observed. Culture anaerobically on blood agar, showing a clear zone of hemolysis. A good anaerobic culture media for cultivation here is the Robertson cooked meat media. Biochemical reactions for identification of Clostridium perfringens. First, fermentation of sugars as glucose, lactose, and maltose with acid and gas production. Then, stormy clot reaction. It is a very important reaction where the organism causes rapid fermentation of the lactose present in the media called litmus milk and to produce gas that split the clot formed. Naggler's reaction. when the organism is inoculated on an egg yolk media, it produces opalescence. This opalescence due to the production of lysosinase enzyme that make precipitate around the colonies. If we add the antitoxin in the media, this precipitate can be inhibited. Treatment and prophylaxis: surgical debridement of devitalized tissue, amputation may be needed, parenteral penicillin and metronidazole administration, hyperbaric oxygen, as oxygen, as we said before, is toxic to the bacilli, passive immunization with gas gangrene antitoxin. Finally, we are going to talk about Clostridium botulinum, which is a causative agent of botulism. Botulism, food poisoning, was formerly associated with the ingestion of contaminated sausage, hence its name. The bacilli is obligatory anaerobes, gram-positive bacilli. Its spores are oval, central, and sometimes subterminal. Its events factors: botulinum toxin is a very potent neurotoxin of 8 antigenic types from A to H. Type A, B, and E are the most common causes of botulinum in man. There are heat labile toxins, and these toxins are the most potent toxins known. The transmission, where the spores in the soil contaminate the vegetables and meat, and when this food is canned without adequate sterilization, the spores germinate due to the anaerobic conditions formed inside the cans and produce the botulinum toxin. And when it is absorbed by the gut, it is carried by the blood to the nervous system, where it blocks the release of acetylcholine at the neuromuscular junction, leading to flaccid paralysis of all muscles. Again, botulinum toxin causes flaccid paralysis of all muscles, while in case of Clostridium tetani, the neurotoxin there causes severe spasm of all muscles, but here flaccid paralysis of all muscles. Clinical signs: the most important clinical sign that is dilated pupils due to the ocular muscle paralysis. The death is usually due to respiratory muscle paralysis. Clinical types of botulism: either food poisoning due to eating the canned food contaminated with the spores, or when these spores enter inside the wound, causing a wound botulism. And one of the most popular forms is the infant botulism. This is due to the ingestion of infant to honey contaminated with the spores. Honey is a favorable environment for the growth of the botulinum spores, and so it is preferable to be prevented in the first year of life. Laboratory diagnosis also made and suspected on clinical ground. Direct detection of the toxin in the food remnants or in the patient's serum. Injection intraperitoneal to the animal, resulting in flaccid paralysis and death. The food remnants can be emulsified and heated at 80 degrees centigrade, and the extract is plated out on the blood agar anaerobically to detect the Clostridium botulinum bacilli. Finally, the treatment is the immediate administration of the antitoxin. Trivalent antitoxin here against type A, B, and E of the toxins, and respiratory support due to the respiratory paralysis. Control: any swollen cans should be discarded immediately and proper sterilization of all canned foods. Thank you.